This is another one of our biochemical tests. This is looking for starch hydrolysis. In other words, what you're going to do is you're going to grow bacteria on a starch agar plate. This is a media where starch is the sole carbon source, and you're trying to see if the bacteria, when they are growing, are able to utilize that starch, break it down, and use it as a carbon source. So you're going to inoculate the plate, and then after growth, you're going to test to see if starch is still present. If starch is still present, then that means the bacteria was not able to use it, and so it would be negative for hydrolysis. It didn't break it down. If starch is no longer present on the plate, then that means yes, the bacteria was able to break down that starch, was able to use it, and broke it down, so there's no starch remaining anymore. It was hydrolyzed. Now, the way you test for this is kind of backwards or reverse from what you usually think. You're going to add iodine. Iodine reacts with starch, and usually when iodine reacts with starch, it will turn a dark color. So to test for starch hydrolysis, the ability to break it down, after growth on the plate, you are going to add a drop of potassium iodine. A positive test for starch hydrolysis is when after the iodine has been added, it's clear or it's very light tan color. That indicates that no starch is present. So this is where some people get confused. It's a negative test for starch. Starch is not present, but then that means why is there no starch there? Because the organism is positive for starch hydrolysis. It broke it down. That's why it's not there. If you add the iodine and there's no clearing and it remains this dark, some people describe it as dark blue, it's almost like a black color. So there's no clearing around the bacteria when you've added the iodine, and it's this dark color. That means starch is present. So that's a positive test for starch, but in this case, why is the starch still there? Because the organism was not able to hydrolyze. It was not able to use the starch. So it would be a negative starch hydrolysis result. So this is just a schematic drawing showing that if you have a plate, a starch plate, the black lines indicate it where there is growth, and over here where there is no clearing and it remains the darker color around it, because when you put iodine on the plate, the starch was still present in this area, so is that very dark color. Yes, starch is present, therefore there was no starch hydrolysis for this particular organism. It would be negative. On this side where you get this clearing and lighter color around it, that in turn would be negative for starch, so it's positive for starch hydrolysis. This is shown on an actual plate of where iodine has been added, and it is still, the iodine naturally is kind of this light amber type of color. And so right around the bacteria over here, there is no starch. So it was positive for starch hydrolysis. Now right here you can see it is darker in color, but you also see there's no growth there. So there's no bacteria. So there is a strip in here where there is still starch present, but there's no growth there. So that makes sense that there's starch there because there's no bacteria to break it down. So for this sample, it would be recorded as positive for starch hydrolysis because it did break down the starch in the area where you have growth.